Hey guys, hi, welcome to Unacademy. This is Unacademy Future Doctors, and I am your educator for biochemistry, Dr. Shivangi Shukla. Here we meet again after a gap of one day. I hope you guys haven't been lazy, you guys have revised in this day. Let me just check if I'm visible and audible to all of you and if the live stream is functioning smoothly. Give me a minute to check whether everything is alright or not. Yes, okay. So we meet after a gap of one day, and I hope this day you have utilized to revise what we have completed. We have been taking very exhaustive sessions, right? I have around 4 sessions per day. Tonight I have 6 sessions, 4 YouTube sessions, and 2 on Unacademy. So I hope that you guys are also working as hard as I am for you, because in the end, it is you who has to take the exams. Okay, so please try and put in a little bit more effort, because this time you can utilize. This is the time where you are not under very much pressure. Also, this is the time in which you're not going around because of the COVID situation outside. So since you have to stay at home, so you might as well study. Okay. All right then. Let us start. Would be knowing about me till now. By now, you guys have followed me for the past 1.5, 2 months now. Okay, even yeah, this is almost 2 months since I have started my classes on this channel. So I'll just quickly take you through the different types of subscriptions for NEET PG available on Unacademy. The Plus as well as the Iconic subscription. Both of these subscriptions will give you access to video lectures from top educators across the country, using different state-of-the-art tools that are available on the app. And the Iconic subscription, in addition, will also give you a subscription to resources from Prep Ladder. Okay, so in these times, Unacademy offers you one of the most subsidized and inexpensive rates that are available in the market. And on top of that, you can get an additional 10% discount by putting in my code DRSHUKLA. Okay, Dr. Shukla. This code is going to fetch you an additional 10% discount over and above the highly subsidized price already. Okay, so please do give it a thought, guys. It is not a bargain. This is not a bargain that will result in a loss. Okay, this is something that is going to help you, and you will be very happy. You'll never regret your decision to go ahead with the subscription, especially in these times where the online method of learning is the only hope for regularity in studies. Okay, all right. So I have for these 2-3 days, what I have decided for you guys is to come up with special classes on this channel where I will try and make you revise different metabolic cycles in a unique manner. How? I will choose different organelles in the cell, and then I will ask you in an interactive one-on-one session here on the channel, and I'll ask you what processes do you think occur in this organelle. Okay, so for today I have chosen lysosome in this session. Okay, now lysosomes are they are very hardworking organelles in the cell. They take part in a lot of metabolic processes. Okay, now see, there is one thing that we know about a metabolic process. Then we should know at which site it is occurring in the cell. Then we should know in which organ or organs it is occurring inside the body. And then we should know how it is integrated with the other metabolic cycles. Okay. So if we read it in a back and forth, different from different multiple dimensions, if we read the same thing, then it is going to open up our avenues, and it is going to make the understanding even deeper. Okay, understood. So where all are these lysosomes used in the cell? First of all, I'll just quickly brief you about what lysosomes are for those of you who don't know. Lyso means break. Okay, lysis means breakage. Okay, to divide, to break. And zome, as I told you, zome is any body. So lysosomes are bodies that cause breakage or division. Okay, so they cause breakage of some biomolecules, or so to say, breakage of some molecules, breakage of some structures, lysis of some structures. Okay, now there are multiple types of lysosomes. First of all, how are they formed? They are bound by a membrane. Okay, they are bound by a membrane. Similar to the cell membrane, they also have a phospholipid membrane. How are they formed? They are pinched off. This is the correct keyword. They are pinched off from the endoplasmic reticulum. Okay, and they undergo processing where? In the Golgi bodies. Okay, understood? So they are pinched off from the endoplasmic reticulum and they undergo processing in the Golgi bodies. Okay. Now, so what will be? They will have a similar structure as the endoplasmic reticulum. Okay, because they are pinched off like this from the sides of the endoplasmic reticulum. See, suppose this is the endoplasmic reticulum. Okay, what does it have? Endoplasmic reticulum has stacks that are called cisternae. These stacks are called cistern. These small individual units in the stacks are called cisternae, and this whole stack is the endoplasmic reticulum. Okay, now what happens is from the sides, by the pinching of the cell membrane, what happens is small vesicles will be pinched off, and these vesicles, they will be processed in the Golgi apparatus. Okay, in the Golgi apparatus they will be processed, and then what will this, after processing, these vesicles will form lysosomes. Okay, understood. Now what are the different types of lysosomes, if you can ask? Can you guys name it? In the comment section, can you guys tell me what are the different types of lysosomes? See, suppose this is a cell. Okay, this is the nucleus, the endoplasmic reticulum. Is giving off, pinching of this, and then it goes and gets processed in the Golgi apparatus, and then it forms this lysosome. Okay, this one is the first or the primary lysosome. What is this? This is the primary lysosome. Now, what is the primary lysosome? This primary lysosome has a lot of enzymes, metabolic enzymes. Like, what are these enzymes? They are hydrolases, nucleases, proteases, different enzymes are there. Okay, hydrolases, hydrolytic enzymes that are used in digestion, proteases, nucleases, peptidases, and the like. Okay, so this one is this one, number one is what? This is the primary lysosome. Got it? Now what happens is this, whenever the cell ingests something, suppose there is a foreign allergen or some foreign body that the cell is trying to ingest, that the cell is ingesting, that the cell has come across. Okay, so this is the foreign body. Now this is coming near the cell membrane. Okay, now what will the cell do? It will invaginate. The cell membrane will invaginate like this, and it will take in this foreign antigen. Okay, now what will happen after it has invaginated wholly? What will happen? this will pinch off from the cell membrane. Okay, this will pinch off like this. And what will be formed? This is a phagosome. Okay, now if this is a water droplet or if this is some kind of a liquid, then what will be formed? If this was some kind of a liquid, then a pinosome will be formed. Okay, understood? I'll mark this here. Are you guys following? So what is this? This is a phagosome. What is phago? Phago means eating. Zome means body. So this body has eaten this food. Okay. This is pinosome. Pino means pina. Pina means drinking. Okay. So pinosome. इसने पी लिया. क्या पी लिया? ये droplet. This body has drunken this. Got it? Now what happens is this primary lysosome will then attach to this phagosome or the pinosome. And what will it form? It will form a secondary lysosome which has engulfed this. The secondary lysosome would have engulfed this phagosome or pinosome. And this has a lot of hydrolytic enzymes also. So what is this called? This is called a secondary lysosome. Secondary lysosome is also called what? This secondary lysosome is combined, is what? This is lysosome combined with phagosome. So it is called phagolysosome. Got it? Secondary lysosome or phagolysosome. Got it? Now what will happen? What does the lysosome have? It has a lot of hydrolytic enzymes. So it will break down this. So this phagosome will be digested. Now after digestion, whatever is left, that will be called, whatever is left inside this body is called, these undigested products, this will form the third type of lysosome, which is the residual body. Why residual? Residual means it could not be digested. It is left behind. So you understood the three types of lysosomes. First is the primary lysosome. Second is the secondary lysosome or the phagolysosome. And third is what? The residual body. Okay. Now secondary lysosome, there will be digestion of the phagosome, of the ingested antigen or the ingested foreign body. Okay, there will be digestion of the ingested foreign body by the hydrolytic enzymes. Okay, and what will form after digestion? This residual body contains the undigested components that could not be digested by the hydrolytic enzyme. Okay, understood? So these are the three stages of lysosome. Understood? I'll take you back to this diagram. It is very, very important. Okay, this is the nucleolus, this is the nucleus. These, all of you should know how to draw the different organelles in an animal and a plant cell. It is very important. So whenever you are asked, because one or the other question you will be asked about some organelle in biochemistry. So whenever you are asked, please make sure that you draw the diagram of the cell. Okay, that much is expected of you. It is something that is of pre-MBBS level. So this much is expected of you. Okay, understood? You should know how to draw different organelles. You should know how to, you should always try and illustrate. You can be asked something about the mitochondria. You can be asked something about the electron transport chain, where you should know how to draw the mitochondrial membrane. You should know how to draw these. Okay, these are expected of you and rightly so. Okay, so you have followed till now. Now should I go further? Now I'll tell you what are the different, now I'll tell you where and all these will be involved. Okay, got it? Just a second. Okay. So now I'll show you where is all of this involved. Lysosomes, uh, the first and foremost thing that we talk about lysosomes is in cell digestion. Okay, so whenever you're talking about digestion, then you would be talking about the different hydrolytic enzymes. Okay, hydrolytic enzymes, first of all, whenever you're talking about something, if you're reading biochemistry, I want you to always focus, always try and click inside your head, or always try and recall inside your head as to what is there something from biochemistry that I can apply here. So if you're reading digestion, then it should, all the biochemical aspects should very clearly click inside your head. Okay, so the digestive enzymes are hydrolytic enzymes. That means what group of enzymes they fall into? Hydrolases. What are hydrolases? They cause breakdown with the introduction, with the addition of water molecule, isn't it? This is the function of hydrolases. Now these are the digestive enzymes. Now they cause digestion. Where are these hydrolytic enzymes stored? Where are they coming from? From the lysosomes. So one very important function is in the digestion. That is, these lysosomes, they release their hydrolytic enzymes. Okay. Now there is something called as autophagy. What is autophagy? Autophagy, auto means same or self. Okay. Phagy, autoimmune means immune system working against the self. Similarly, autophagy means self, and phagy means, as we have seen in phago, phago means eating. So phagy means eating, self-eating. What will this do? Here the lysosomes, all the primary lysosomes, they don't have any foreign phagosome. There is no foreign phagosome with which the primary lysosome is combining. Here, multiple primary lysosomes will combine. Multiple primary lysosomes will combine together. Okay, like this. And they will now destroy the cell debris, the all the used up products that are used up products of metabolism that have accumulated inside the cell. They will release their digestive enzymes to metabolize or to recycle that debris. Now whatever biomolecules can be recycled from that are taken up by the cell, by different organelles, and they can be used again, and the rest of them are converted into residual bodies and excreted. Now how is residual body excreted? This residual body will, like this phagosome, it will go and join towards the membrane like this. Okay, and it will be budded off through the process of exocytosis. So similarly here, when this autophagy happens, the cell debris is removed through exocytosis after digestion in the primary lysosome. Now, autophagy we have heard, we have understood. Now, what is autolysis? Phagy means eating. Lysis means breaking. Auto means same. So, autophagy is self-eating, eating up the cell or the contents of the cell. Autolysis means breakdown of the whole cell, breakdown of the cell. Okay, this is because of this autolysis, lysosomes are called the suicide bags of the cell. Now, whenever the cell has to be degraded, whenever the cell has to be destroyed, why? Because there have been a lot of toxins that have developed in the cell, and now there is a lot of wear and tear in the cell, and it can either be due to necrosis or due to apoptosis, that is programmed cell death. Then what happens? The lysosomes, they burst. They burst and they release. This is the lysosome. It will burst like this. And it will release its digestive enzymes like this, the hydrolytic enzyme. Now this is the animal cell in which it is present. These hydrolytic enzymes are going to digest the whole of the cell and the cell will be destroyed. Okay, what is the example of autolysis? Like, have you seen the lizards? They, whenever they are frightened, they drop off their tail. So that is the process of autolysis, through the process of autolysis. Okay, now suppose this is the lizard. Okay, now this is the lizard. Now what happens is, whenever it will be scared, what will this do? The lysosomes present in this part of the body that joins the tail with the rest of the body, they are going to burst inside their cells and these cells will die. And these cells will die. This tail is not no longer connected to the body and this tail will be dropped off. And now this what will this do? It is a protective mechanism by the lizard. Now this the tail will fall down and the predator will think that this is the lizard, and the predator will go after this tail and the lizard can quickly escape. Okay, so this autolysis is a protective mechanism in most of the organisms. Understood this? Got it? Now let us recall all the things that we have read. Okay, in the conversion of procollagen to collagen, in the conversion of proinsulin to insulin, what did we read? That the pro group is eliminated. The pro group contains some extension peptides, isn't it? Extension peptides. Proinsulin contains the C peptide. Procollagen contains what? What does proinsulin contain? I'll mention. I would have felt happy if you guys had mentioned by yourself. Procollagen contains the amino extension peptides that are present at the amino terminal containing intra-chain disulfide linkage, and the carboxy extension peptides present at the carboxy terminal containing intra and interchain disulfide linkage. Okay, now they have to be broken down, they have to be removed for self-assembly of collagen, isn't it? Please go back and watch that lesson on extracellular matrix and on collagen. I have discussed this time and again. Okay, so they have to be broken down for the self-assembly of the triple helix of the three chains of the three helices of the collagen. So they are broken down by aminoproteinase and carboxyproteinase. And what forms is the collagen molecule through self-assembly of the tripeptide of the three chains. Okay, so these aminoproteinase, carboxyproteinase are present where? Where are they coming from? From the lysosomes. They're all proteinases present in the lysosomes. Similarly, proinsulin. What does proinsulin have? Proinsulin has the C peptide or the connecting peptide. This C peptide is present as a connection between the B and the A chains. Okay, this C peptide is removed by hydrolytic enzymes again, which are secreted from the lysosomes, and upon removal of the C peptide, B and A peptide are joined together to form the mature insulin. Understood? So this is the practical application of what you have read about lysosomes. Now again, one more thing. In the secretion of thyroid hormones also you have read. Have you read about this? Thyroid hormones, where are they formed? In the follicular cell. Follicular cell. What are the two thyroid hormones that we have read about? T4 and T3. Tetraiodothyronin and triiodothyronin. This one is lesser active. This is more metabolically active. T3 is more metabolically active. Now they are formed in the follicular cell. They are stored in the colloid. How are they stored? First, what happens is the iodide transporter concentrates them against their concentration gradient inside the follicular cell. Then they move towards the colloid where they are oxidized. Then they iodinate the thyroglobulin molecule. The thyroglobulin protein has a lot of tyrosine residues. 115 tyrosine residues are there, which are iodinated by the iodine ion, okay, to form monoiodotyrosine, diiodotyrosine, monoiodotyrosine, diiodotyrosine. Now they will couple together. All this is happening in the colloid. Now they will couple together. This diiodo is 2, monoiodo is 1. Mono means 1, di means 2. It has 2 iodine, it has 1 iodine. So this 1 plus 2 will become 3. MIT DIT will couple to form T3. DIT and DIT will couple to form 2 plus 2, T4. Okay, and there will be some T3, some T4, and some inactive MIT DIT will also remain. Okay, now this thyroglobulin that contains this thyroid hormones and the immature and the inactive DIT MIT is stored inside the colloid. Okay, now whenever TSH acts on the thyroid gland, what happens is the thyroglobulin molecule is released. Where is it released? This is the follicular cell and this is the colloid. Okay, this part is the colloid and this is the follicular cell of the thyroid. Now this is a thyroglobulin molecule containing T4, T3, MIT, DIT, T3, T4. Now TSH has acted upon it. Now it will be engulfed like a phagosome. Okay. Now this has been engulfed like a phagosome. It has come inside. Now what will happen? Phagosome has come inside. Now what will come? Our dear hero primary lysosome. This will come into the picture. This primary lysosome will combine with this phagosome, which is thyroglobulin molecule, and it will form the secondary lysosome. This secondary lysosome will then release its hydrolytic enzymes and move the T4 and T3 from the thyroglobulin. So what happens? T4 and T3 are released from thyroglobulin. Osteopetrosis. What is osteopetrosis? This is also called bone within bone. It means the bone is very, very dense. Okay. It is so dense because there is no or very minimal clastic activity happening and all osteoblastic activity is only happening. So what will happen? The bone is, okay, in osteopetrosis, the bone that is formed is extremely brittle. Okay, and this happens due to defective resorption because of defect in lysosomes. Discussed a lot about lysosomes. Followed. Okay, just let me check if everything is clear. Give me a second to check the comment section. If there's any doubt, then you can ask me. So this is the basic function of the lysosomes. Now we can start with, in the next session, one very, very reticulum is the synthesis of cholesterol, and that we're going to revise. Okay? Understood or not? Let me just check if